first edition of the 2022 CCAIM seminar series. My name's Andreas Floto. I co-direct the Cambridge Centre for AI in Medicine, and it's my great pleasure to introduce David Ascher, who some of you will know from his time working with Tom Blundell's group in biochemistry in Cambridge, where together they pioneered really kind of exciting transformational approaches to using machine learning to predict the impact of mutation on protein function. And since then, David has continued with his stratospheric rise, currently at the University of Queensland, where he's going to tell us what he's been up to in terms of, I think the title is Unravelling the Molecular Mechanisms Behind Mutations and Their Link to Phenotypes Using Graph-Based Signatures. David, thank you again for coming, and over to you.
Thank you very much for the kind invitation, Andreas. I should, if I've got privileges, share my screen. There we go. Hopefully you can all see that. So thank you for the kind introduction. I'm sorry it's a slightly awkward time for you guys, because I know you normally do it later in the afternoon, but with time zones the way they are, it's sort of what fitted the best. I'm going to take you through a little bit about what the group's been working on over the last nearly 10 years now, and where we've been trying to apply that. But feel free, if there's things that interest you more than others, I'm happy to sort of tailor things as we go and spend more time on certain areas. So our group, it's broken down into three sort of broad main research streams. The two I'm going to be talking about today is primarily what I started on with Tom Blundell back in Cambridge. So that's where we're trying to use a structural genomics approach in order to better guide the clinical decisions and drug discovery. And that marries up really closely with some of our chemoinformatics approaches, which I'm not really going to go into much today. We've also started up a more recent area around clinical informatics and trying to combine genomics and structural information together with clinical data to improve things. But we'll focus mostly on structure today. And so what I'm going to do is I'm going to take you through a bit about how we've been developing the methods. And in particular, hopefully I'll cover some of the limitations of the methods, because as I'm sure you guys are all aware, a method's only as good as the data it's trained on. And we're dealing with a very biased subset of data that has been published and is available. So there are inherent limitations within the methods that we develop that you need to take into consideration when you then start looking at trying to apply it to different situations. And then I'm going to take us through two different areas where we've been trying to apply some of these approaches, namely around human diseases and overcoming drug resistance. So to start things off, one of the big ideas within the group has been how can we get from genotypic information through to the phenotype, and whether that's a human or whether that's a bug or a virus, the links aren't always very obvious. And a lot of previous approaches or a lot of established clinical tools very much relied upon primarily looking at the DNA change and trying to associate that with a phenotypic output without considering a lot of the intervening biological information that takes place within that. So two of the most popular tools for characterizing DNA variations, or certainly when we started, were Polyphen and SIFT. And what you see is that there's a very small overlap in the mutations that are deemed as either benign or pathogenic between, there you see three different approaches, so SIFT, Polyphen, and mutation taster, where if there's obviously a lot of congruence between the methods, you've got confidence that you've probably got a pathogenic mutation or a benign. But what do you do if one method out of the three says something, or two of the three? So there was a lot of divergence, and these were primarily machine learning driven methods. So they are inherently biased themselves based on the data that they were being used to train them. And what we knew was that SIFT and polyphen in particular were trained on a large proportion of pathogenic mutations. So clinicians, and as a general rule, like scientists, will publish positive data as opposed to negative data. It's very hard to get out there and say, hey, look, I looked at this mutation, I've characterized it experimentally, and it doesn't do anything. Most of what you see in the literature is, hey, we found this mutation, we've done some experiments, and hey, look, it's got this wonderful effect, or very bad effect, depending on what way you want to look at it. So the inherent data that they were using at the time was biased, and that led to a bias in the methods themselves. And a number of papers came out around that time where essentially they were showing that a high proportion of the mutations that were being classed as pathogenic really weren't pathogenic. Or if they were, they certainly didn't have a high enough penetrance to really be defined as pathogenic in most people. So we needed better ways to try and interpret this genetic data. And so what we came up with, the hypothesis was, but if we went down that idea of the central dogma of science, you go from DNA to RNA to proteins, those proteins interact, those interaction networks are in larger pathways, and how all of these things interact together ultimately leads to the phenotype we see. If we can move further down that biological process, we can incorporate more information that can hopefully better guide our interpretation of novel variants. And so, of course, coming from a structural background, we started off looking at protein structure to start with. And so the most simple mutation that you can possibly get is a single amino acid change within the coding region. And they can have very, very subtle effects that make it hard to predict. So this is where we've focused a lot of our attention on. You know, when there's large insertions and deletions, of course, that's going to have a very large effect on the protein activity or function. But if we can predict what's happening when you just change one single amino acid across the entire protein, it gives us a lot of insight, particularly when we're looking at very small changes, often within our patient samples. And if we think about what can happen within the protein structure, it can change how that protein folds, it can change its stability or its conformation dynamics, it can change its degradation, its interactions, how it's modified, its activity. So there's a lot of things it could do. How can we predict that? What can we do with that information? And how can we do it in a very high throughput fashion, was where we started to approach this from. Now, going back a step, one of the students in the group who's recently finished, Michael Silk, was working with Slave Petrovsky, who's now up in Cambridge with Ascher, on a score that Slave had developed called the missense tolerance score. And it's a way of measuring purifying selection within a population. So it's looking at the ratio of observed to expected variation within a given region. So you can look across the whole gene, but you can also look much smaller than that. And what Michael and Slave showed was the intolerant regions, so regions where you don't see the amount of missense variation that you would expect to see, are highly enriched in pathogenic variants. Which makes sense, right? It's where we're seeing a lot of dominant effects. But when we start looking at that within a protein three-dimensional structure standpoint, what we saw was that a lot of these intolerant regions were clustering in three-dimensional space. So they weren't just necessarily clustering within a one-dimensional window within the sequence, it was clustering in 3 dimensions as well. So we wanted to be able to try and capture some of that information. And so what Michael developed was a missense tolerance ratio that actually considered 3-dimensional space rather than just a window at the gene level. And what he showed was that considering structural information, his ability to differentiate pathogenic and benign mutations was as good as SIFT or PolyPhen without needing any additional training. So this is just using purely population information. So that's the spatial MTR there. And so this has really spurred us down that pathway as well. It's all really well and good to consider things at a sequence level, but we really do need to take into account protein structural information. And so where we started was actually somewhere where Tom started many, many years ago. So Tom developed a method called site-directed mutator, which was probably the first method for predicting the effects of mutations on protein structure and protein stability. And the way he approached that at the time was using substitution tables, so evolutionary substitution tables. So it's not just saying what's the chance of an alanine mutating to a tyrosine, it's what's the chance of an alanine mutating to a tyrosine if it's on an alpha helix buried within the core of a protein. And so we worked with a really talented postdoc at the time, Arun, to redevelop that while I was in Cambridge and update it with more recent information. And for a purely statistical approach that isn't taking the wealth of experimental data out there to refine, it actually performed quite remarkably well, which for those of you who've worked with Tom, probably shouldn't come as too much of a surprise. But there was still significant room for improvement. So we're getting correlations in that 0.4 to 0.5 range. We wanted to sort of see what can we do to really push that beyond. And we started to branch into machine learning. And so a postdoc that was working with Tom at the time, Douglas Perez, had developed something during his PhD called a graph-based signature. So it's a way to represent the three-dimensional structure of a protein. What we do is we take each atom as a node. The interactions between those atoms are the edges, and we look what atoms and interactions are within a given window around point of interest, in this case, a mutation. And we can essentially represent that as a matrix of numbers that's very easy for a machine learning algorithm to interpret, as opposed to a PDB file. But beyond just capturing the geometry, we also needed to capture some of the physicochemical properties. So we included what we've called a pharmacophore, which is a very simple way of representing the mutational change that's occurring within that mutation. And so, of course, we started at the same place that Tom had started, and we applied this to looking at the changes in protein stability. And we developed the first of our tools, which was called MCSM stability. And what we saw was when we applied it to independent blind data that neither SDM nor MCSM had seen, it performed reasonably well. So it performed better than SDM, which was encouraging. And what we liked was even more than that was when we combined information from both the structural information, so our graph-based signatures, with Tom's original SDM approach, the statistical-based information, we saw an improvement in overall performance. So it sort of highlighted to us the importance of considering not just structural information, but also sequence information. So the two are complementary, and you can't start ignoring one or the other. But I mentioned that there are inherent biases within the data. Well, most of the data that we were using to train MCSM is on very nice soluble globular proteins. It's what people can work in the lab very easily on. So it represents a large proportion of the data that's been published and put out there. Membrane proteins are a special case. Obviously, they are not soluble, they're not easy to work with, so they weren't very highly enriched within the data set. And what we saw was that across all the methods that we really tested out when we had a nice data set on just transmembrane proteins, none of the methods that had been developed for looking at protein stability effects within membrane protein, within any protein, translated well to membrane proteins. And you can sort of see that reflected in the correlations there. So the highest one there, which is TMP, was built on the data that we used to evaluate this. And you can see that even it's only getting a correlation of around 0.5. So we used a transfer learning approach to develop MCSM membrane. So MCSM membrane never saw the transmembrane data, or at least this set of data, but we used the information within our original data set on transmembrane proteins to tweak our algorithm and develop something that was specific for membrane proteins. So again, we were able to eke things out and get a performance boost, but it comes back to always thinking about the data that, well, if the method was never trained or designed to build on that data, then is it really going to be applicable? And you can see right down the bottom there, STM didn't perform particularly well on transmembrane proteins at all. And again, I think that's reflected in part by the nature of the substitution tables that we were using. Another limitation within the data, and this comes back to the issue with SIFT and polyphen I mentioned, most mutations that people publish have an effect. You don't tend to publish things and say, oh, it was neutral. Most of the time what people are publishing are mutations that either destabilize and significantly destabilize, or they show some level of increase in stability. So if they're doing some protein engineering type approach. But even if you think about a purely random mutagenesis experiment, if you mutate most residues in the protein, you're probably going to destabilize it somewhat. You're still not going to get very many mutations that lead to an increase in stability. So you see that sort of a bias within the data set we were using. It was very skewed. And so we had a look, and again, all of the programs that were designed to, particularly machine learning programs that were designed to predict the effects of mutations, performed quite poorly on stabilizing mutations. They captured destabilizing ones quite well. And the best one there on the stabilizing was SDM, because it didn't have that inherent bias from using a machine learning approach. So we needed a way to overcome this. And so what we did was we went back to what Tom had originally proposed in SDM, which was a thermodynamic cycle between the unfolded and folded wild type and mutant states. And within a sort of a closed system, the broad hypothesis is that the change in Gibbs free energy of going from a wild type to a mutant should be the negative of going from the mutant to the wild type. So it's essentially the inverse. It's not going to always hold true, but it certainly is a way that we can use to try and balance out this data a little bit more. So what we did was we modeled all the mutants and we termed that our wild type, and we used the information on the wild type as the sort of de facto mutant to try and balance out this data somewhat. And so a talented PhD student in the group, Carlos, used this as a way to try and develop a more balanced machine learning algorithm. So you can see his two attempts there, Dynamute and Dynamute 2, which show much better performance across stabilizing mutations, which is very encouraging. The other thing Carlos did, which was quite nice, was we had only ever really been using static structures at that time. You know, here's a model of a protein, or here's the crystal structure, but we know proteins are very dynamic. And so he used an NMA, so a normal mode analysis approach, to try and better capture protein dynamic information as well as structural information within his methodology. And so one of the questions that I'm always asked, and I think Chandra's asked this one a few times, is, okay, so this has all been trained on experimental structures. How does it perform on homology models? And so one of our master's students, Sunny, who's just started his PhD in the group, went back to answer this question in a very systematic fashion. So he developed homology models of proteins that we hadn't used in our experimental training sets, and evaluated, I think, 10 different methods plus a few different sequence-based methods to try and look at the relationship between the sequence identity of the homology model to the performance of the different predictive tools. And what we see is probably what we'd expect to see across most of the methods. We see a decrease in performance as you drop sequence identity. If we sort of draw a rough threshold across all of the programs, it's somewhere around that 30 to 40% mark, which I sort of, from a structural biology perspective, it sort of makes sense. But in the process of doing this, or while he was doing this, a new method came out, of course, in 2020 must have been, AlphaFold 2, which sort of completely redefined the whole landscape of homology modeling. So Google's attempt at this was sort of light years ahead of what we had been doing at the homology modeling standpoint. And so what Sunny did was he went back and he reanalyzed all of these methods using AlphaFold 2 structures instead of traditional homology modeled structures. And what we see is that the AlphaFold 2 performs pretty much on par with the experimental data. In a lot of cases, it actually performs better than using experimental structures, which is interesting in and of its own right. And it's something we haven't really delved into too much. But it sort of opens up now the opportunity that, well, with DeepMind and AlphaFold, applying AlphaFold to pretty much every single organism in the UniProt over the next year or two, we're going to be able to use these methods on any protein structure that they've developed with a degree of confidence that it should be replicating what we have seen previously. But I've spent a lot of time so far, and I haven't really sort of advanced you beyond just looking at stability, which is, you know, something Tom had been doing 20 years ago or more. So one of the areas we really wanted to get into was looking at protein interactions. So we know Mike Sternberg's group has shown a few times that between 40 to 60% of disease-causing variants are located near protein-protein interaction interfaces. Yeah, that's a large number of mutations that we're just not going to capture very well if all we're doing is considering protein stability data. And that's just looking at protein-protein interactions. What happens if we look at drug binding sites or ligand binding sites or other functional areas of the protein? The number of mutations there that we would be missing and important information that we'd be missing sort of grows exponentially. And so Carlos, again, has gone back to look at how we can use the AlphaFold 2 models to try and capture some of these interaction data in a better way. And two of the limitations of the AlphaFold 2 models is that it's only APO. So it's sort of, here is a structure, and it's a blank canvas for you to do something with. And they've only looked at canonical transcripts. So for the humans, that means you're missing out a lot of potential transcript information. So the canonical transcript, that's sort of easy to fix. We've gone back and now used AlphaFold across over 140,000 human transcripts. So we can sort of build up a data set now of the entire human transcriptome. But it still didn't help us with the question of, well, this is just sort of a blank canvas. How can we get this in a high throughput way, some of the interaction information? I can certainly do it at a modeling perspective or a docking perspective, but trying to do this high throughput across an entire data set, or every single protein in the human transcriptome and then other organisms, is a much more complicated task. So what Carlos has done is he's built a method that uses sequence and structural homology to place ligands in our APO models based upon known experimentally solved structures. So it doesn't capture everything, and we know it doesn't. We're still trying to work on that, but it certainly is a way that we can, in a quite high throughput fashion, automatically populate out our APO models to make them more useful for some of the things we're interested in looking at. And so this is a method that he's sort of just been working on over the last few months. So it's just hitting the press, and this is the first time and the first group of people who will have heard about it, which is quite exciting. And so then taking that structural information of all the interactions and just applying our same graph-based signature approach, we've then looked at the effects of mutations on protein-protein interfaces, nucleic acid interactions, small molecule interactions, and metal ion interactions. So you sort of see that we're trying to build up a bigger picture of all of the potential molecular consequences that could happen if you have a coding mutation. Again, it's always nice to start evaluating your data and looking at it in different perspectives. So while we were developing some of the PPI tools, Capri was running a competition at the time. So Capri is a very, very powerful and useful, I'm not sure what to call it. It's not a resource, it's a program essentially where people can submit predictions on experimentally designed data sets that have never been released to the public. So it's a way to try and evaluate what methods are really going to perform and how generalizable they are to new data sets. And so Carlos did this for two of his methods, so MCSM PPI2, and it performed pretty much on par with what we would expect to see from our training data set, which was quite exciting. We were able to avoid some of the issues with the bias in the data sets between mutations that lead to an increase and a decrease in binding affinities by using the hypothetical reverse mutations again. And then another data set bias that we hadn't thought of, but another PhD student in the group, Yu-Chan, looked into us for, was that a lot of a lot of the mutational data that people were generating or have generated is alanine scanning, because it's a very easy approach, particularly at a protein-protein interface, to try and identify hotspot residues. So it's very, very enriched in mutations to alanine. But quite promisingly, we don't see a bias across our data sets and across our methods for mutations to or from alanine. And I think part of that's to do with having used those reverse mutations. So for every mutation that has been to an alanine, we've now got mutations from alanine to something else. So I think we've captured a lot of that we might have otherwise missed. Of course, another bias is when people are trying to mutate things to look at the effects of mutations on an interaction, you tend to make mutations close to that interface. So most of the data tends to be clustered, within 6 angstroms of interaction. And we do see a small decrease in the performance of our methods as we go further away, which is just something for us to sort of work on over time. So pretty much we know that anything beyond 10 angstroms, our methods aren't particularly designed to handle. And in part of that is because the maximum sphere that we use within our graph-based signatures tends to be around 10 angstroms. So beyond that, not only is there less data for us to train and develop the methods, but we're also not capturing some of those long-range interactions within our graph-based signatures that we otherwise might. So it's an area that we're still working and improving on. Another special case are antibodies. So one of the hypotheses that Tom and I had many years ago with a PhD student, so Harry Jubb, is that to a certain extent, antibodies interact with a protein almost more similar to a small molecule than sort of a typical globular protein interaction. And Yu-Chan really saw that when he applied methods that were designed to look at protein-protein interactions to data sets of just antibodies, that their performances had really deteriorated significantly. And so he developed 2 really nice methods that took as a basis the MCSM PPI, so our protein-protein interface, but tailored it specifically for looking at antibody interactions. And we were able to get a really nice improvement in performance across both experimental structures, but the homology test sets. And as sort of a nice bonus, we've been able to apply that to sort of real world data. So we were working with a group in Europe who was trying to engineer an antibody to target tick-borne encephalitis. And they found and identified a antibody that worked really, really strongly against the Siberian strain of the tick-borne encephalitis. So, KD's in the low micromolar range. But it didn't work so well against the European strain. So one of the questions at the time was, well, can we predict mutations using that structure that might change that binding affinity and improve binding to the European strain? So we proposed a few mutations at the interface that we thought might be interesting and could improve affinity. And so one of them there in particular in bold had a marked effect on both the Siberian and the European strains. And we were able to get the Siberian binding affinity down to submicromolar and the European, recognition of the European strain down to the low micromolar, so the 1 micromolar range again, which was a really nice sort of example of how you can apply these methods in a sort of predictive and in a protein engineering way. And again, I've only sort of focused on protein structures at the moment, but we sort of tried to go beyond this to consider, well, what happens within broader networks? So we can model protein-protein interaction networks and metabolic networks as a graph as well. And we had a really nice paper in PNAS a few years back where we used that to try and model what we thought would happen across a metabolic pathway. And we were able to predict with reasonable accuracy the proteins that we thought would be affected by different metabolic stressors or ER-type stressors with quite high accuracy, which was quite promising and something we're, again, developing further to improve our platform. So we're sort of at the halfway mark now, so I'm going to focus a little bit more on where we've been trying to apply these methods and primarily the successes we've had, but I'll also talk about some of the failures we've had along the way as well. And to start things off with, we're going to start looking at how we've been using this within human genetic diseases. But before we do that, just want to recap that what we've developed is a platform to predict a range of different molecular consequences of mutation. But the phenotypes themselves arise from a combination of these different effects. So it's very hard to just say, well, you've decreased the protein-protein interaction. Is that going to be disease-related or not? Is there a way we can try and combine all of that? So one of the things I built when I was with Tom, actually, still, so we had a visiting postdoc from China who helped us out with this, was a pipeline for trying to combine these different predictive tools into a single output. And I don't think you need to worry too much about the details here. The sort of broad scope of it is that mutations that have very large effects on one of those key areas, so protein stability or dynamics or a key interaction, tend to be have a very large phenotypic effect, which is sort of what you would expect. And those that have sort of mild effects across a few different ones, it's how those mild effects interact together that ultimately lead to whether that has a large phenotypic change or not. And so we've been applying this in a more high throughput fashion with 3 different organizations now. It originally started off with a impromptu meeting in Brazil, actually. So Sergio Penna was talking just before Douglas and myself at a conference we were at in Brazil. And afterwards, we chatted over coffee and got talking, well, maybe there's a way we can combine what he's doing at sort of that large genomic screening level within Brazil to try and better improve his ability to identify causal variants. And so when Sergio or most genomic centers analyze a genome of someone with a suspected genetic disease, they'll often identify a lot of different variants that could be causal. Now, it's really easy if that's already characterized variant. If it's already been seen in a human before. It's been characterized in different experimental models. The clinician has to have a reasonable certainty that, well, we've seen this, we know it's associated with this phenotype. We can probably say it's the causal variant here with this patient. But in a lot of cases, there'll be 10, 20, 100 different variants of uncertain significance and no sort of key gene or variant that they think, oh, this is the one we know is and has been characterized before. And so this is where we've been sort of then coming in to apply our structural tools. And so we've been using our in silico approaches and now our AlphaFold fleshed out transcriptome to try and better understand what we think is going on here. And so far across 3 organizations we've been working at, we've analyzed about 160 different patient data. So they'll range anywhere from 5 to 100 variants of unknown significance. And of those that we've sort of said, look, we think these are the causal variants and that have then gone to be experimentally validated, we're getting around 85% congruency between the experimental data and our in silico predictions, which is, again, really exciting. It sort of shows the potential impact that we can have within the genomic sphere by better incorporating structural information. But a lot of what we've done has been more at the a single disease level and saying, well, we want to do more than just say, is this causal variant? Well, what's the penetrance risk of it? What's the actual real-time risk to the patient? What's the potential treatments we can infer from that? And a lot of this started with work we did with Eamon Ma at Addenbrooke's Hospital. So we've worked on a few projects with Eamon, but one of the nicest ones to present today is on a disease that he's quite passionate about, von Hippel-Lindau disease, which is associated with the development of clear cell renal carcinoma. And we developed a model to try and predict, using the structural information, whether we thought a mutation would be benign, so not really have an overall effect, or whether it was the patient was at a low risk of developing renal carcinoma or at a high risk of developing renal carcinoma. And so one of Eamon's students at the time did a very nice study where they took those predictions and looked within their patient cohort. And what they saw was the patients that we said were low risk, we never saw develop clear cell renal carcinoma. So that's sort of consistent. And those that we classed as high risk, a large proportion of them had developed clear cell renal carcinoma within the course of their analyses. So it was sort of a nice way to say, well, structural information doesn't just give you sort of a gross phenotype of a yes/no answer. We can start to make things more granulated and try and get more refined predictions there. And so one of the other projects that we were working on with Eamon was on the succinate dehydrogenase complex, which is mutations in this can lead to glioblastomas. It's a tetramer with so 4 different subunits bound together. It's got metal ion binding sites, it's got substrate binding sites, different protein-protein interactions. So there's a lot of potential consequences a mutation could have. By using that pipeline, we have been looking at both the penetrance of mutations within the succinate dehydrogenase complex, but also patient survival. So if they do develop cancer, what's their survival time? And what we see again is that mutations that have a very large effect on a subunit, so changes in Gibbs free energy over around 1.5 in stability, for example, lead to worse outcomes and higher penetrance, which gives us, again, a nice way to go beyond just a yes/no to try and delve a bit further into what we think the real world impacts of this could be. And another disease we've been working on is called alkaptonuria. It was one of the first metabolic diseases. I think we're running out of time, so I'm going to try and go through this one a little bit quick. And because I want to get to drug resistance, which I know Andreas is quite keen to hear about. So mutations in that lead to alkaptonuria are in a protein called HGD. It's involved in tyrosine metabolism. It's a very beautiful dimer of trimers. It's this nice donut sort of formation. And what we see is that mutations can affect it, be found in the active site, so affect the actual activity of the protein. They can affect how the different subunits fold, but they also can affect how those subunits come together and interact. So again, this one was again with Tom. I took these into the lab, so I experimentally tested out 24 different disease mutations and our predictions on them. It's one of the first times I've ever worked on a set of mutations where I was able to express and purify 24, or a large number of mutations where all of them are known to have a phenotypic effect. Normally, you know, if these are leading to a decrease in stability or the formation of the hexamer, you don't get very good yields, but here we were able to get reasonable yields of all of the proteins. And this was used as part of a way of analysing patient data. And so we're still working on this a bit further with groups in Liverpool and in Slovakia. And one of the interesting things that one of our collaborators in Slovakia have looked at is that mutations that have mild effects tend to have mild outcomes in patients. And those that have large effects, again, have larger effects on patients. So for this, they were looking at the amount of the HGA that was present in urine. And the mutations that had the largest effect, the patients tended to accumulate and excrete the most HGA, indicating that they are progressing worse than patients with mutations with mild mutational effects. And I'm going to skip over this, but we were able to design a more stabilized version of HGD, which we've been using in a as an enzyme replacement treatment, which has been quite exciting. Actually, we're able to get quite significant increases in stability just by including 2 mutations. And in animal models of that, we're seeing some quite promising effects so far. So that's quite exciting to see. But now I want to talk a little bit about drug resistance in the last 10 minutes or so, because that's the other big area we've been sort of working at within that sort of how can we apply this mutational data. And we started off looking at TB. Tom has a very long history of TB, so I think that was somewhat fortuitous for us at the time. But there was also a very pragmatic reason. So drug resistance in TB isn't a result of horizontal gene transfer. It's a result of point mutations generally within coding genes. So it very much replicates what we've been doing and looking at within the human genetic level. It's not that these methods don't apply when there's horizontal gene transfer, and we've done a lot of things looking at penicillin resistance on plasmids and that. It's just TB makes a very nice model because it's not undergoing those horizontal gene transfers. And so as a PhD student in the group, Stephanie Portelli was looking at resistance in 3 key genes that were the targets of either first or second line treatments. And she was doing this in collaboration with the London School of Health and Tropical Medicine, so with Tani Clark and Nick Furnham there. And what she saw is something that we've seen with genetic diseases as well, in that mutations tend to have quite diverse effects depending upon the complex they're in. But most mutations tend not to be found at the active site. Well, within our genetic diseases in particular, but there's a lot of mutations that drive resistance and drive genetic diseases through changes in stability and protein-protein interactions, not just does it stop my drug binding? And with RPOB in particular, Stephanie built a model that was able to predict drug-resistant, whether a mutation would be resistant or not within RPOB, with quite high fidelity, which was really encouraging, because the molecular diagnostic test for rifampicin resistance focuses on just a very small region within the RPOB gene called the RRDR, whereas her method could look at mutations anywhere within the RPOB gene. And working with Sandeep at the leprosy mission, she was able to show that that method translated very, very well to other model organisms. So she could take something that she'd built on TB and apply it to other gram-positive and gram-negative bacteria where there was data on resistance for RPOB mutations. And it had very, very similar levels of performance to what she'd built on using TB, even when the sequence identity was quite low. That was encouraging. The other thing that she observed, and it ties in very close with the story I was telling with the genetic diseases, the mutations that had the largest structural effects tended to have the lowest frequency within the data sets we were studying. So what we saw was that the most prevalent single point mutations tend to be those with mild structural effects. And that can mean that they only lead to mild resistance, but certainly that's a level of resistance better than wild type. And for those mutations that had the largest structural effects and potentially the largest effects on drug binding, they tended to need the presence of compensatory mutations to shift that protein fitness landscape across and make them more favorable. But then taking some of the work that Steph had done and trying to build it within a larger context, what we did was we had a look at a whole pile of TB-resistant mutations throughout the genome. And we tried to build or use our previous pipeline to build a model that to try and differentiate between resistant and benign. And so what we saw was about 80% of mutations tended to be within that 6 to 8 angstroms of the drug binding site. And using our predictive tools, we were able to capture a large portion of those. But there were some very interesting mutations that were found at protein-protein interfaces that led to resistance, which we were able to capture. And very, very few, at least for drug resistance, that affected protein stability. So this is sort of the inverse of what we saw with the genetic diseases. And CatG itself is actually a really fascinating example, because what we saw, or what we know, is that CatG has undergone a gene duplication in TB at some point. So there's two proteins in TB that perform very similar reactions, and they can compensate for each other. But only CatG seems to be involved in INH activation. So mutations that sort of eliminate out CatG don't have as huge a fitness effect on the bacterium as what you might see with knocking out an essential protein, because there's another enzyme that can sort of compensate for that process. But you tend not to see too many highly destabilizing mutations in the drug targets, because if you knock out a drug target with a drug or with a mutation that's just completely destabilizing, you're sort of having that same effect that you're not really, that you're going to have a large fitness cost associated with it. And again, we were able to capture most of that data. And so overall, we were quite happy with the level of performance and ability to differentiate these mutations. But what we then wanted to see was, well, what happens if we apply that elsewhere? And so we took this model, and without retraining or doing anything to it, we applied it to data sets of drug-resistant mutations for cancer, HIV, and a few other viruses. And we achieved very, very similar levels of performance, which was really, really nice to see. And more recently, we took that exact same model and we applied it to looking at drug resistance in plants. So I did my honors on the branch chain amino acid biosynthesis pathway with Luke Cadet many years ago. And we went back to have a look at some of his drug targets there, and we were able to predict with really nice affinity or accuracy mutations that were leading to that could lead to drug resistance against the HAS inhibitors. And it performed as good or better than previous methods that were being developed to look at AHS resistance around dynamic-based type approaches. Now, what can we do with that information? Well, if we can predict a resistant mutation, can we use that to better guide patient treatment? And that's something that a PhD student between myself and the Victorian TB program was looking at, so Malacha Carmarker. And she was focused on a drug called pyrazinamide. So it was a frontline therapy against TB. It was considered essential because it was the only drug that worked in one of the very specific life stages of the TB. But it required an acidic environment to work. So what we found, or what people had found, is that the drug resistant screening assays can be very irreproducible or highly variable because the conditions are so sensitive and hard to deal with. TB itself is a bit of a pain to grow because it's so slow, but you add in the need for acidic media to get the drug to work, and you are adding additional complications there. It was also not a really good target for a molecular diagnostic approach because the mutations, both resistant and susceptible, that people had characterized and seen, were located across the entire protein. So you can see a lot of the protein structure there is in cyan because both resistant and susceptible mutations have been located at those residues. And it's not really conducive to sort of being able to very easily identify what was going on. So what she did was she asked the question, well, can we use the structural effects of mutation within these proteins to try and predict what mutations we think are going to be lead to drug resistance or not? And she was able to do that again quite well, and quite excitingly for her. So this was sort of the first year of her PhD. With Justin, she was able to take that through into the clinic, and they used it to change patient treatment. So it's sort of nice example of how you can go from someone just starting in the area to getting a method into the clinic and actually having a real world impact within quite a short time frame. And so we're now exploring this with other organisations to sort of say, well, it's used in Victoria, can we try and do larger studies? So in particular with FIND, they were looking at, can we apply this within their American-based studies that they were running? The other thing that we've been quite interested in is, can we use this information to preemptively predict resistant mutations during the drug development pipeline? And so while I was working with Tom, we had a couple of very interesting collaborators. So the MM4TB collaboration and the HIT-TB collaboration have both performed phenotypic screens and come up with hits against the same protein. So GuaB2 or IMPDH is involved in guanine biosynthesis. And Andreas will probably remember this story because at the phenotypic screens they were doing, these compounds worked really well, but they ended up not being great targets for the clinic because when we moved them to animal models, they tended to have much lower efficacy, in part because of the high levels of guanine present in the lungs. It meant that it wasn't the essential protein that it was within the phenotypic screen. So it's a nice example of why you should always be very careful about using purely phenotypic screens. But one of the things we did was we applied our models to try and look at what we thought would be likely drug-resistant mutations within the IMPDH structure. And we identified one in particular that was interacting with some of the structures that postdoc had solved, so Angela. This tyrosine 487 was a very strong candidate for us. It wasn't involved in protein-protein interaction, so it could easily be mutated without affecting the tetrameric structure of IMPTH. Mutating it didn't seem to affect protein stability. It didn't seem to have a large effect on binding of the natural NAD, but it had a very large effect on binding of the compounds that were being studied. And so Valerie Miserati was running some in vitro screening assays, and compounds that we found interacted very closely with this tyrosine developed resistance very quickly, and those that didn't develop resistance very quickly. So it's sort of a nice example that, well, we can try and use some of this structural information in that drug development process to hopefully design and develop better drugs. So what I'm terming resistant drugs. And so now we're trying to combine all of this into sort of a broader platform where we can take information from fragment screening, virtual screening approaches, and combine it with both our structural insights, but also some of our chemoinformatic insights around medchem and PK/PD properties to try and make drug development a more streamlined process, or at least reduce the number of failures we need to have along the path. And one of the places where we've been applying this more recently has been in COVID, but I think what we'll do is we'll wrap up here so there's time for some questions. Hopefully I've convinced you about the importance and the interest of using structural information to better understand genetic variations, which leads me to thank all of our collaborators and funders and the group in particular. I'm very lucky to work with a really great group of people. You can see the newest member of the group there actually. So my daughter Ari made it to our Christmas party last year, not last year, the year before. And thankfully she stayed asleep for this whole talk. And I'm just going to leave on this slide. So all of our tools that we've developed are freely available. Please feel free to use them. If you've got questions about using them, don't hesitate to ask. The only reason we bother developing them is hopefully that people use them and find them useful. So ultimately, it's up to you guys to utilize them within your frameworks as best as possible. So thank you for the time and the opportunity, and happy to answer some questions.
David, thank you very much for an excellent presentation. There are so many things, so many questions coming out. So can I just remind everyone, please post them in the chat if you've got any questions. Let me start off with a question for you, David. I just wonder what your thoughts were about the opportunities to look at the failures of your methods in order to A, refine the machine learning approaches, or B, to indicate which types of experimental data you would need to generate more of in order to improve function?
Yeah, a lot of that has been by trial and error as we've gone along. Some of the times I've discovered things afterwards and I thought, well, why haven't I considered that before? One of the probably more obvious examples was when we first built the nucleic acid interaction predictor. We lumped DNA and RNA together, and as soon as we'd published this, I sort of thought, why did we do that? And we noticed an enormous improvement in the performance of the method as soon as we separated it out into single-stranded RNA, double-stranded RNA, double-stranded DNA, single-stranded DNA. As soon as we sort of broke it down into those subsets, we noticed an enormous improvement because you're capturing that information in a more balanced fashion. Mm-hmm. So a lot of the time, it's not always obvious when we've done it, but we always try and sort of feed that information back. The other thing we haven't really gotten a chance to go into in this, but sort of a pet peeve of mine is I really don't like black boxes. So all of our machine learning algorithms, we try and make them interpretable to try and understand why the method is predicting this is something. I never like it when it's sort of a black box and people throw in a whole pile of things, some of it which doesn't even make sense, and they get out an answer and it's like, well, why does it say that? Yeah. And I think a lot of the time during our development process, we often find that a very informative way. So I term it within the group, though that's probably not a correct term, knowledge-driven machine learning. So we try and do it in two ways. We do it both while we're developing it, but also we take back those failures to try and continue to refine things.
Great stuff. Good. So there's time for a few questions from the chat then. So Andrew Baldwin writes, have you compared against your methods against co-evolutionary fitness predictors such as EV mutation?
Yes, I believe Sunny might have actually done that already in his. The performance seems to be, again, it depends what you're looking at, what protein system, what mutations, but our methods perform as good, if not better, most of the time.
Good, that's clear. Okay, question from Randy Reed here. Interested in your opinion on CAGI, so critical assessment of genome interpretation, which is coming soon. Do you take part in these challenges? Do you think they're informative for benchmarking methods to predict the impact of VUSs, variants of unknown significance, I should translate?
Yes, yes to both. I think, yes, we do take part. We haven't taken part in as many as we would like to have, and that's more just resources and time, because it does sap up time in a group without necessarily always getting a publication. So you can't always justify it for, say, a student. But I do think they're very valuable benchmarking tools in general, because I think what we've seen experimentally is you don't trust papers, right? Just because someone says, hey, I've developed this really cool assay, it doesn't mean it translates to another lab or another situation particularly well. And I think industry has learnt that the hard way. So I think there's a lot of distrust with both computational and experimental methods out there, that how reproducible are they all? I think things like CAGI give us the opportunity to show, well, hey, look, when you take brand new data that no one has seen, these methods really are performing the way they are. And I think AlphaFold is actually a really nice example of where these sorts of tests work out really well, because when people saw how AlphaFold worked on their test, it sort of just lit up on social media, right? Everyone knew that this was going to be a huge thing way before their bioarchive preprint, way before the method became available. And I think that was a really nice, powerful way that they could actually demonstrate the utility of their method.
Great stuff. Question from Aaron, Environment. Thank you for a great talk. People have attempted to use AlphaFold 2 directly to assess impact of protein stability or PPI directly, but this doesn't seem to work very well. Do you think it's a promising avenue to modify these deep learning architectures to achieve this nonetheless, or will these always be two separate problems and will require specific tools like MCSM?
Absolutely brilliant question. So at the moment, no, it doesn't capture it very well, but it isn't what AlphaFold was designed to capture. So I don't think it's a fair test on AlphaFold to say, hey, look, it doesn't work. It's not what it was ever built to do. So it's sort of like a of course it hasn't really worked the way it has. It's still nice that people have tested it, but because, you know, AlphaFold wasn't designed to look at protein-protein interactions, but it was able to be adapted to do that really, really effectively and well. I have very little doubt that at some point people will be able to predict the effects of mutations using a deep learning architecture like AlphaFold. I think there are a couple of complications though. One is data availability. So when you look in the PDB, a lot of the point mutations that have experimental structures have very, very similar experimental structures to the wild type. It's not surprising, right? There's a confirmation bias there. It's been able to be experimentally solved. It's probably not that highly disruptive in the first place. So I think there's a data issue that they need to get around in order to be able to do it. The other thing is that even though AlphaFold does really well at modeling structures, a lot of the low confidence regions, so the disordered regions, they're just sort of placed in locations, right? What do you do if a mutation completely unfolds the protein? What does it output? Does it outfold just a spaghetti? Does it try and fold? So I think, yes, I can see eventually something happening, but I don't think it's the same method. And I think there needs to be more data available or different data to make that more of a powerful resource. I think it's also worth saying, particularly with someone like Randy around, that AlphaFold was only successful because of the last, 40, 50, 60 years of experimental structures that have been solved. There's a vast amount of experimental data out there that they were able to leverage in order to develop their methods. And I think at the moment that data is not really available on the mutational front to
so you're going to stay in business for a bit then, Dave?
Hopefully.
Well, listen,
we know they're working on it, right? We know that they are working on it, but I don't think it's a 1 year, 2 year problem.
Got it.
I could be wrong.
Well,
listen, it's on YouTube now. Everyone's going to hold me to it.
Exactly. Well, listen, we're well over time, but it was a terrific seminar. David, hopefully we're going to see lots more of you in Cambridge as we develop our collaboration with Cambridge Centre for AI and Medicine.
I hope so.
Yeah, and really looking forward to seeing where this all goes. So thanks everyone for their time. Just to remind you that we'll send out the links to the YouTube recording of this, and hopefully David can answer some of the remaining questions in the chat later. But thank you, everyone.
More than happy to.
Okay.
Thanks, Andreas.
Great, David. Brilliant. Yeah.
Oh, thank you.
All right. Cheers. Thanks.
Thank you, David. That was brilliant.
No, my pleasure. I was just starting to have a look at some of the questions that were coming in during it. I really loved it.
It was really good.
I think
people were very
engaged.
Really good there. Yeah.
That's it. So Andreas mentioned answering the questions.
Yeah.
How do I do that? I'm guessing most people have gone by now.
Yeah,
I suppose if they want to ask, they can email me.
Yeah, exactly.
Yeah.
Okay. Navneet is on the phone. Okay. Yeah, no, that's great. And people can always email us. So if we get any questions by email, we will just send them your way.
Well, the other thing is those that are there, you feel free to unmute yourself. Ask now. I've got what, 20 minutes or so still.
Mm-hmm.
Hi, sorry, I'm in a public place, so there might be some background.
It's okay. I've got a daughter sleeping who could wake up at any moment and start screaming. So I think either way we're fine.
Right. So I guess quickly, what about your opinion on if the data is all biased towards pathogenicity, then how do we train our models? This is the first question.
Very carefully. I mean, there's a lot of ways that you can there's a lot of different ways that you can deal with biased data, right? You can try and unbias it, which is what we did with the hypothetical reverse mutations. You can do that by oversampling or undersampling. You can do it by adding weights. So there's a lot of different ways that you can try and better capture it. I think the other side of it is also the quality of the data. So, you know, as an example, if you're using ClinVar variants, do you just take the highest confidence ones, or do you start delving down into some lower confidence, you know, 4 and 3 based stars? So I think there's different ways you can go about it without necessarily automatically just saying you need to collect more data. But I think it's something that a lot of people, when they build these methods, don't always consider, and they don't always consider the real world implications of it.
Right. So, sorry, there's a follow-up to this, slightly different question. When you observe a particular mutation, it might not be pathogenic at that stage. So, for example, if a patient is 30 years old, the mutation might be benign, but at 70 or somewhere ahead, it might be pathogenic. So, what are your thoughts on accounting for that?
Yeah, so that comes back to some of the stuff that we've been trying to do around going from just a yes, no, so pathogenic, non-pathogenic, to some of those more nuanced approaches where we look at rates of survival or penetrance. Particularly when you're looking at, say, cancer-based mutations, it's not one mutation that leads to cancer, right? It's an accumulation of mutations over a lifespan and how they all interact. It's more that penetrance and the probability of it developing at some point. And I think what we still see is what we'd expect, that mutations that have the largest effect on proteins, you see occur the penetrance and the development of the disease earlier in the lifespan. But it's, I think it's one of those things that is harder to do on a whole genome level. It needs to be done on a disease level, on even a protein level.
Okay, thank you.
That's okay. Happy. I think there was someone else.
Hi, yeah. I'm still here.
Hey, Andrew.
I think I asked a question in the chat briefly about how FEP could potentially be used to confirm or confirm or test some of those drug-resistant mutations.
Yeah.
Although I think actually it might be more interesting to talk about the MTR stuff. I don't know if your answer is brief on that.
Look, I think the brief thing is, yes, people can. Yes, people have. It's the same thing also with the mutational stuff. I've seen people trying to use MD-based approaches to then validate their mutational predictions. I think the gold standard is always still going to be experimental data.
Yeah, absolutely.
And that's where we've tried to go back to. I think this also goes partly to Ascher's question as well. People have also tried using both FEP and MD to increase their data set sizes. So, you know, you've got a naturally biased, like naturally limited data set. Can we use some of these more computationally intensive approaches to generate more data that you can then train more high throughput things on using machine learning? I think we've seen mixed successes with those so far. I don't think that's saying that it absolutely won't work, but again, I think it's always being careful, and having those experimental data sets, I think, is always quite powerful to go back to and say, hey, look, how is this actually performing real world? Yeah,
definitely. But yeah, no, the MTR 3D stuff is very interesting. For reference, I'm a PhD student in the chemistry department actually at Cambridge, and I've been working on well, some stuff that's very similar to the MTR 3D stuff, specifically related to regional sort of constraint.
Excellent.
So
well, one of the things I didn't even get a chance to mention is we find that population-based constraints is more informative than evolutionary.
Yeah, this is part of the reason I'm sort of trying to think about it, because there's such a massive divergence in some genes and their function.
Exactly. And it's the big assumption whenever you're doing an evolutionary alignment that every protein in that alignment is the same protein. Okay, with careful curation, you can be reasonably confident with that, but also that every single protein is under the same restraints. So it's forming those same interactions, the same residues are important across species. And that's okay for something that is, I'm not going to say ubiquitous, but is for an interaction that has occurred across evolution. But for something that's developed more recently, you don't get those same strong signals. And there was some nice work out of Canada looking at PPIs between Cerevisiae and Pombe. So you've got two yeast, right? You would expect the PPIs between them should be reasonably well conserved. But I think they only found 40% of protein-protein interactions were conserved between the two.
Wow, that's crazy.
A large number that if you were to do an alignment between them, you're going to not capture some of that. Whereas population-based information, well, protein X in me, you, and everyone else is, in theory, going to be under the exact same restraint. So we see those signals come through better.
Yeah, I guess it's a reminder that there's lots of potential biology out there. I read an interesting paper actually about the gonadotrophin releasing hormone receptor, which has actually become destabilized in like some mammals. If you compared the mammalian receptor to the chicken ones, you'd see, oh, these are loads of mutations which must be fine because they're in mammals, right? But no. Do we want the protein to be less stable?
I think this is also where it was not always obvious when you're looking at a protein, or when you're looking at a closed system, what real world impacts that is. So a nice example more recently is the Omicron variant. You immediately think, oh, okay, we look at the binding, they see reduced binding to ACE2, I think it is. And if you're predicting that, you think, oh, that should bind less well. It should be, potentially less virulent. But no, actually is more virulent because it gets into different areas of the body. And it's very interesting that what you always think might happen isn't always what does happen.
Yeah, I think this sort of the approach of like carefully considering each of the possible outcomes, and having to do that essentially in a semi-manual way, but using that knowledge guiding machine learning is a good way of dealing with that where we have the knowledge. So I'd be, if you're considering updating MTR3D at any point, I'm not actually sure how. I didn't get to see how recent it was, but there's new data coming out about that all the time, I think.
So we published MTR3D last year.
Okay.
So it's got all the variation from UK Biobank, Nomad and things like that, and a few other biobanks that we've pretty much, I think there's 600,000 individuals in total we've got there. But it's one of those things, the more you get, the more powerful it is. The other thing is it's still a very basic score. So one of the things we're trying to do with it is also take into account frequencies. So at the moment, we just look at, well, is there a mutation there or not? It's sort of a binary thing, but it can become a lot more nuanced if you start looking at the frequencies of those different mutations and comparing the frequencies to the expected frequencies within those spots. That I think has a lot of potential as well as we get to these larger numbers.
Yes, yeah. I think you had to be, I've been thinking about this actually. I think you had to be very careful because of like all sorts of genetic drift if it's realistically neutral. And some of these mutations might be basically neutral reproductively, if not, actually. But what are your thoughts?
That's another bias, right? You look at the data sets, they are white male professors, and I mean, not all 600,000 of them, but certainly a lot of the early genomic sequencing was a very limited sample size. And we're trying to get beyond that, but it's still biased. It's still not as representative as it should be. I don't know if your focus is human or bacteria, but we actually did a really nice one that we haven't published yet, where we applied the MTR approach to malaria. And because there's a lot of genomic sequencing on malaria. And what we see is there's actually a really nice, so I talked specifically about regions that are depleted of variation, because that's sort of linked to human diseases. But with malaria, what you actually see is regions enriched with variation. So rather than having MTRs that approach 0, they have MTRs that approach 1.5 or even 2. And pretty much all of malaria's drug-resistant mutations tend to be located in those regions. So
that's really interesting to see.
Yeah, it's nice. It's hard to sort of do it in an unbiased fashion though, because just there's so much resistance in malaria already that it's sort of present within the data sets. But it would have been nice if we could strip out all resistance, looking what those regions would have looked like otherwise.
Yeah. Oh, that's cool. No, my specific interest is in human GPCRs. But in the same way that you've got like well, some regions which are, you know, particularly important ligand facing, and some which aren't. And I think that's what I'm hoping to look into.
And GPCRs, I mean, even more complicated than that, right? Because you've got the allosteric sites and everything.
Yeah, absolutely. It's
we've only sort of dabbled on the GPCRs so far.
Yeah, I'd be interested in talking more at some point.
Happy to.
Yeah.
Feel free to shoot me an email.
Great. Yeah, nice to meet you.
Nice to meet you too.
All right. Bye for now. Thanks for the
Andrew.
talk.
Thanks, Ascher.
Thank you.
So I think they were the
they were the last ones. Yeah.
But you always know the keen people, right?
Yeah, exactly. No, and that was good to have some extra addition.
Excellent. Well, thank you again for the opportunity.
No, thanks for doing this. It was great having you. Really good talk.
It was fun. Yeah, do feel free to share my email or pass along any questions that people have, because I'm sure there'll be stuff in the chat that we'll miss, but